Okay, let's dive right into this explainer. Today, we're unpacking the fast-paced, hard biological reality of how Matrixyl and Matrixine peptides actually go about rebuilding your skin. We're pulling directly from a brilliant peptide proof report, and we're strictly sticking to the science, the clinical data, and the literal cellular mechanics of what these ingredients do the second you apply them. So no marketing fluff today, just the data-rich reality.
Right off the bat, to understand why any of this matters, we have to look at the brutal math of human aging. Take a guess how many collagen fibers your skin loses every single minute. 30,000. 30,000. Now, when we're younger, the body replaces most of that. But the minute you hit 25, that replacement rate starts slipping. And by the time you're 50, you are losing collagen way faster than your skin can rebuild it, creating this massive structural deficit. You literally cannot just hydrate your way out of this. You have to find a way to tell your skin to restart its building program.
And to reverse that math, we have to look at a very specific subset of ingredients called matrikines. Now, matrikines are not just random protein fragments or cellular waste. They are highly active messengers derived directly from extracellular matrix proteins like collagen and elastin. When your skin structures break down, these fragments trigger biological activity. They literally bind to cell surface receptors on your fibroblasts and act like a 911 call, signaling that damage has occurred and new collagen is desperately needed.
Section 1. The accidental messenger. Let's look at the origin story here, because surprisingly, this massive chemical signaling breakthrough wasn't actually born in a cosmetics lab. Picture this. It's the early 90s, and researchers at the University of Tennessee are doing clinical wound healing studies. They're just watching how the human body repairs tissue after an injury. Well, they accidentally discover that collagen breakdown products naturally act as a distress signal. They isolate the key fragment, a 5-amino acid sequence known as KTTKS. But there was a major catch. KTTKS is water-soluble, meaning it's way too large to slip through your skin's outer lipid layer on its own. Enter Sederma. Their team ingeniously attached a palmityl fatty acid chain to the peptide, making it lipophilic enough to finally penetrate the skin barrier. They patented this resulting molecule as Matrixyl in 1993, and it was an absolute game changer. Moving to Section 2, cellular construction. Let's zoom in to the cellular level to see exactly how this molecule issues a biological work order. The mechanics here are so elegantly straightforward once you understand the biology. Step 1. Palmityl KTTKS crosses into the dermis. Step 2 is where the magic really happens. It activates TGF-beta. Think of TGF-beta as the ultimate master switch for collagen production. It acts exactly like a site foreman at a construction site shouting, "Pour the concrete." That leads us directly to step 3, where fibroblasts ramp up gene transcription and start pumping fresh pro-collagen into the extracellular space. Your skin sees this peptide and simply obeys the command to rebuild. And this brings us to why this particular peptide is unusually complete. A 1993 paper in the Journal of Biological Chemistry proved it doesn't just boost structural proteins, it actually stimulates glycosaminoglycans, or GAGs, and raises hyaluronic acid levels. It's essentially pumping up the gel-like ground substance of a healthy dermis. So you aren't just getting more structural protein, you're getting a much more hydrated, resilient matrix at the exact same time. Section 3, unpacking the clinical evidence. Because it's one thing to see this work in a petri dish, but proving this mechanism works on actual human skin, that's the crucial part. The most cited clinical trial on Matrixyl dropped in 2005 by Robinson and colleagues. This was the gold standard, a 12-week, double-blind, placebo-controlled split-face study on 93 women. They used a cream with just 3 parts per million of palmitoyl KTKS, and the data? A statistically significant 17% decrease in fine lines compared to the placebo, alongside better skin texture and dropped photo damage scores. But guys, here is the hard biological reality that skincare marketing departments conveniently ignore. Those improvements took 8 to 12 weeks to appear. Let me repeat that. Nobody saw results in the first month. The effect built gradually and finally plateaued around week 12. Why? Because you are fundamentally rebuilding the extracellular matrix. You aren't just temporarily plumping wrinkles with surface hydration. True biological tissue remodeling absolutely requires time. And we know for a fact it's actual tissue remodeling, thanks to a smaller 2005 study by Osborne. They went deep and took literal punch biopsies from treated skin. Under a microscope, they saw measurably thicker collagen bundles and an increased density of fibrillin-rich microfibrils. These microfibrils are the crucial anchors at the dermal-epidermal junction. When they degrade, your skin starts to sag.
This histology data proves Matrixyl actively helps restore those exact anchoring structures. Section 4. The Matrixyl Evolution. Let's see how this initial breakthrough turned into an entire franchise engineered to target distinctly different matrix proteins.
So Cedar Ma obviously didn't stop with the original formula. While the first Matrixyl targeted bulk dermal collagen, they leveled up with Matrixyl 3000, adding a second matricin fragment, palmitoyl GHK. GHK has copper-binding properties, which lets the formula hit collagen production and tissue remodeling from two angles at once. Then we get the latest generation, Matrixyl Synth 6. This one uses palmitoyl tripeptide 38 to hyper-target the basement membrane. It literally anchors the dermal-epidermal junction by stimulating 6 completely different matrix components.
Because Synthesis 6 focuses so heavily on those deep anchors, rather than just bulk collagen, its clinical profile is incredibly unique. The data shows it drops wrinkle volume by roughly 20% over 8 weeks. But the real standout result? Dramatic improvements in skin firmness and elasticity. It's essentially tightening the biological fasteners that hold all your skin layers together. Section 5. Signal amplification and the myth of more. Now, I know it sounds crazy that such incredibly low concentrations, literally 3 parts per million, can achieve macroscopic structural changes. But it all comes down to a fundamental rule of biology. It's a biological process called signal amplification. A tiny trigger sets off a massive receptor cascade. One single peptide molecule reaching a fibroblast doesn't just produce one single collagen molecule. No, it triggers a cascade that commands the cell to churn out thousands of collagen molecules over several days. You have a very small input yielding a disproportionately massive biological output. Which, naturally, brings up a highly provocative question that completely nukes a common marketing myth we see everywhere in the beauty industry. If a tiny trigger works this well, shouldn't a serum with 10 times the peptide concentration work 10 times better? The scientific data gives us a resounding no. Because these cellular receptors totally saturate at roughly 5 parts per million, adding more peptide beyond that threshold is biologically useless. The receptors are full, the cell is already maxed out. Knowing that a huge chunk of skincare marketing relies on this more is better philosophy, it really leaves us with one final thought. How many other expensive skincare claims out there are simply defying the strict laws of molecular biology?